Thanks everyone for joining. Welcome to our annual cancer research update with Dr. Annette Khaled. This is always a very important thing that we do every year. We are raising money for cancer research, and Dr. Khaled and her research is why we do it. This is also a really important year for us. 10 years of the Cure Bowl, 10 years of bringing teams together to find a cure for cancer. So really appreciate everyone joining us. I know Dr. Khaled shared, she's got a lot of updates for us today, so don't want to take up too much time. So we will go ahead and turn it over to Dr. Khaled.
Well, I am really excited to be here. I always look forward to this presentation every year. Maybe we should do it twice a year, actually, with the amount of stuff I have to show you guys today. But I titled it today, The Tricks of Precision Medicine for Cancer. And I want to say that the funds that we received from you all this year, or last year to this year, I've been using to do some very expensive research, because we've now been probing into the molecular level of what our therapeutic target, which I'm going to talk about, which is called TRIC or CCT, there by the title, and hopefully share to you what we've been learning this year about how our therapeutic approach actually fits with this concept, precision medicine. So as I was share my screen and get started. And please feel free to stop me anytime during the presentation. I'll try to give pauses every once in a while if you have any questions. Happy to address them anytime during my talking. So don't wait to the end. When I was preparing this presentation, one of the things that occurred to me is I really never talked about what is cancer. And if you Google this, you'll get a lot of different information. But essentially, we actually don't really know, despite all the work that we all do, what actually is cancer. We know that we have normal cells in our body that are functioning perfectly fine. And suddenly, for reasons that we still don't quite yet understand, some cells change and become cancer cells, and eventually over time can kill us if we're not treated appropriately. But this is kind of a little overview of what is cancer, at least to give you a perspective of where we're heading with what we're doing in my lab. So you can see the top of this figure, you see normal cells. Well, what happens to all of us, and this has happening in our bodies all the time, is that we get these mutations in the genes that are found in our cells. So the genes are basically the blueprints that make the proteins that do all the work. And the genes that are the blueprints get these mutations, these alterations. And what normally happens is that if a cell gets too many mutations, that cell is destroyed by our body. And our body basically is surveilling and making sure that no abnormal cells continue on in our bodies. But what happens over time is that we have this cycle of mutations in our genes that are not repaired or removed. And as we go from youth through middle age to adulthood, and then to old age, as we transition through the years, we start accumulating multiple mutations in the same cell. And unfortunately, these multiple mutation process that occurs over time can lead to forming a colony of these abnormal cells that eventually form some sort of solid mass, for example, if it's a solid tumor or a blood cancer, that we call cancer. And that's the point that we actually detect the cancer. We don't detect the cancer during this process of years of these gradual mutations occurring. We detect it when it's actually formed some sort of lump or solid tumor in our body somewhere. And sadly, what kills us at the end, if we have cancer, is when that cancer spreads to other parts of our body and actually destroys our normal healthy tissues and replaces the normal healthy tissue with cancerous tissues. And we lose the function of vital organs like our lungs or our liver or our brain, things like that, that get affected when these cancer cells take over. So that's in a nutshell what is cancer, what we know about cancer. But there's so many gaps in our understanding of this process that I just want to give you start an appreciation of our knowledge base actually about cancer, despite all the discoveries, is still, we don't quite really understand what it is and why it happens. But when we do detect cancer, how do we treat it? So for most patients, what we do is a traditional cancer with the treatment, which is chemotherapy, which is some sort of poison. Truthfully, it's a toxin or poison that destroys not only the cancer, but also the cells in the body. Radiation, which has actually gotten much better over the years. They've gotten much better at actually directing this radioactive, lasers or waves to the tumor sites and minimize the damage to neighboring healthy tissue. But it still can happen. Or surgery, which is basically removing, if it's possible, the actual tumor itself. And that's doable if the tumor hasn't spread too far, where removing the tumor actually destroys healthy tissue. But this is what most patients do. Where we want to go and where the field is really heading is a development of what are called precision medicine. So this is traditional medicine, what I just talked about. Precision medicine is trying to find what were those genetic mutations that I just talked about a few minutes ago that accumulate over time that produce a cancerous cell. What were those genetic mutations? Identifying them and then trying to connect that genetic mutation or those genetic mutations to either a drug or treatment. So it's genetics tied to medicine, and that's what's called precision medicine. And those can include immunotherapy, which has really been a great boost in the last few years of treatments where when it works, it's actually extremely effective. It's almost a cure when immunotherapy works. And then targeted therapies where it's finding that drug that matches the genetic mutation in the tumor and linking the two together. But in reality, so this is precision medicine. It's getting information about the tumor, what the tumor is composed of, what were the mutations that cause the tumor, and then connecting that to either a treatment, a diagnosis, or a monitoring scheme for that patient. So that's the idea. However, this is kind of the scheme of how it happens. You start off with some sort of screening or early diagnosis. This is not precision medicine yet. This is kind of still, doing a mammogram if you have testing for breast cancer, PSA test for prostate cancer, things like that. This is that screening part to try to detect cancer early. But if cancer is detected, usually they'll do, as I mentioned, chemo, radiation, surgery, some sort of treatment for that patient. But here's where the big problem comes. You may get an initial treatment where it mitigates the tumor, right? Either removes the tumor growth is reduced or something. But for many patients, these tumors come back. And they come back either as a residual that was left over that didn't get eliminated by the initial treatments, or it's new growth, new mutations that occurred over time, and you have new growth driven by new mutations. But you can end up with advanced stage disease. And this is where the precision medicine idea comes in, because what they want to do is, when you have this now recurrent cancer, to find the cancer and find some sort of genetic marker, biomarker, that is tied to some sort of treatment. So you can see a little blue panel here where we have the advanced stage disease. This is an example of lung cancer. They had 3 different nodes of cancer in the lung. They biopsied those nodes, and they will identify maybe some treatments based on what the genetic markers that they found in those cancer nodes were. But unfortunately, this approach is not a cure. Cancers will come back. They will evade this approach and arise from new tumors or new mutations. And then you have to do this whole process again. Find another marker, tie it to another drug, and do this process. So it's almost like a vicious circle of trying to do this. And eventually, people run out of drugs because we don't have enough drugs, and I'll talk about in a second, to really do this, to be able to match mutation to drug. So bottom line, for precision medicine to work, you've got to match the right patient, right treatment, and right time, which is actually very difficult to do. Last year, moving into this year, there has been 17 new cancer drugs that have been approved by the FDA, which on the surface sounds really exciting, right? We got 17 new cancer drugs, right? Well, actually, when you start to look at what these drugs are, and this is a couple of them, they're new drugs targeting old targets. In other words, they're just a better form of an older drug. So drugs like toripalimab, which is a PD-1 inhibitor. PD-1 inhibitors have been around since 2016. And receptor tyrosine kinase inhibitors have been around since 2006, 2008. So over 20 years, those have been around. So they're new drugs, but they're targeting old targets. Or existing drugs that maybe were used for one type of cancer are now being used for another type of cancer. So it's a new indication. this is generally what is moving the field. And what we don't have are the new drugs and the new targets. Unfortunately, in terms of precision medicine, only about 14% of cancer patients actually undergo this process of biomarker and precision medicine, and about 7% receive benefits. Now, these numbers are improving and they're getting better, but just giving you a sense, it's still a very small fraction of all cancer patients that are actually able to benefit from precision medicine. Partly because the genetic mutations that cause cancer are not druggable. Many of them are just very complicated protein products that come from these genetic mutations, and we can't find drugs that inhibit them. So that's one problem. And the other problem is, in fact, most cancers are driven by genetic defects for which there are no drugs available. So we have to start from scratch building those. And this is really the clincher. 95% of cancer drugs that are being currently tested in human clinical trials never get approved. Only 5% actually make it over into use of patients. So this is where the field is at. So there's a lot of room here for new discovery. And that's what my lab is focusing on. We are focusing on trying to address this problem and add to the field a new approach for cancer treatments. So, Me Too drugs, I just talked about those. Those are drugs that, new drugs, the same target. This is the problem. There's a slow growth of what are called first-in-class. And I'm going to use that term throughout my talk today, so I'm going to briefly explain it. What a first-in-class drug is, it's the first time this drug has ever been tested in humans. It's been tested in animals, for example, mice, which we do in my lab, but it's never been tested in humans. This is where the potential for growth lies, in the discovery of first-in-class. And actually, I was reading a paper a few days ago where they were talking about that a couple of years ago, right after COVID, there was actually a big increase every year of more first-in-class cancer drugs going and being tested. But that's been tapering off now. So there's been a slowing of growth of these first-in-class drugs. And bottom line is, as I explained to you with the cycle of biomarker testing and then matching to a drug that they do with patients that have advanced cancers, this approach doesn't cure. This approach basically gives the patient more, extends their life, which is of course very important, but it's really not a cure. And importantly, they're very expensive. This process is actually extremely expensive with very, very high price tags. So it's very hard for all the patients to be able to afford to do this precision medicine approach. So for all these reasons, this is where we're at right now in the field. This is where the field stands today. We have cancer heterogeneity, which basically means that everybody's cancer is different. And even everybody's cancer over time changes. So there's a lot of variability. Cancers are very unstable. If you just treat everybody's individual cancer based on their genetic defects, you can treat only a very small percentage of people with the existing drugs that we have. The vast majority of people with cancer don't match to a drug. And that's the reality. So how do we solve this? Well, this is where I'm excited to tell you, because we're taking a little different approach. We're doing precision medicine, but we're doing it from a different perspective. Right now, the field is all focused on DNA. DNA has the genes that get mutated. They produce this abnormal RNA, and they produce this abnormal protein that causes cancer. So what actually causes the cancer is the protein that comes out of the mutated DNA. Well, that's been our focus. We're not focusing on the DNA. We're focusing on the proteins that come from the mutated DNA. So our idea is bypass genetic testing. You don't need to do genetic testing of tumors. Go straight to treatment. The minute you have a detection of a tumor by whatever diagnostic test you're doing, you can go straight to treatment without having to try to match your genetic defect to a treatment. That's our idea. So how we're going to do this is by targeting something we call protein folding. And so a little bit of background with what is protein folding. So I just talked about DNA. DNA is the blueprint. It's going to tell the cell how to make protein. And there's an intermediate kind of translator between the DNA and the protein, which is called RNA. So RNA basically takes the DNA information, translates it into a form that the cell can recognize to make proteins. So proteins, when they're first made from the RNA, don't have any shape. If you think about it, it's like a long string of beads, like a little figure here, has no function, doesn't do anything. It actually has to be folded into a very complicated three-dimensional shape. And that process needs energy, which energy is provided in the form of something that's called ATP, which is basically the currency of energy that all cells need. All cells operate, that's their fuel. All cells operate on ATP. So you need ATP to fold proteins. And you have to move almost like if you think about stability, you have to move from an unstable form to a stable form. So you have to kind of move from one form to another so that you go through these intermediate steps. And that takes time if you didn't do something. So if you just let this process naturally happen from an unfolded to a folded state, that would take so many hours that the cell would not be able to function. So timing is really critical in folding. So all these elements come into protein folding. So what we've learned in my lab and others is that cancer cells themselves are highly dependent on protein folding. They need protein folding even more than your average normal cell does because they are so genetically unstable. They're constantly producing unfolded proteins. So this dependency on protein folding is a vulnerability. It is a point of intervention that we can do as a treatment approach. And that's, again, where my lab is heading. So who does the protein folding in a cell? Well, there's this complex of multiple proteins that in a whole it's called a chaperone. But there's three major bodies of proteins that are involved in helping proteins fold, go from unfolded to folded. There you have your heat shock proteins, your cofactors, and your chaperones. And heat shock proteins have actually been studied as targets for therapy for many years, almost 20 years now. And they've never gotten through clinical trials successfully. Any inhibitor of heat shock proteins fails in clinical trials. And the reason is heat shock proteins are among the most abundant proteins any cell in your body has, not just cancer cells. Every cell in your body, most abundant protein are heat shock proteins. So it's very hard to find a therapeutic window. In other words, you really have to give patients such high doses of a heat shock protein inhibitor to have it be effective that it doesn't work. So these guys are out. They don't work really as a good therapeutic. But chaperones have not been studied at all. In fact, my lab is one of the few labs in the entire world that studies chaperones in cancer. There's very few of us. But they're actually really good targets for cancer. And I think I showed you last year, I didn't include the slide this year, how every year there's a few more publications every year that come out on chaperones in cancer. And we're among the people who are adding to that field of studying chaperones. So I study one, it's called CCT. So CCT, here's a picture of it. It's called chaperone-contained TCP1. It has two names, CCT or TRIC. I like to call it CCT because it's cute, but either name, you'll see that in the field. And what it is, it's like a barrel. It's composed of 8 different proteins in a 2-ring structure. You can see here the top view. If you looked at the chaperone from the top, you'd see this hole going through the center. That's where unfolded proteins go inside. It actually has an iris that opens and closes with energy. And when the iris closes, that drives the folding of proteins. And when the proteins are folded, they pop back out. So kind of showing you how the chaperone works. And I'm going to talk a little bit about one subunit, which is called CCT2, that we found is among the most important for the function of the whole complex. So we don't have to study 8 different subunits. We just study one subunit, CCT2, and that reflects the activity of the whole complex. So just keep that in mind. But I found this really cute little video to explain how chaperones work. So I'm just going to run through it real quick and then get to my data. But I thought just to show you how amazing nature is. I mean, every time I look at this type of stuff, it just amazes me how intricate and involved our bodies are. So a chaperone, think of it like a little nurse, right? So it's a nurse found in every single cell of your body. And it has energy, and that's what these 3 little P's are that look here. This is ATP. So it actually carries its fuel with it. So it has ATP attached to it already. So its fuel is there. When it finds a protein that is misfolded or unfolded, when a protein is not properly folded, it has these areas that are called hydrophobic. And these are areas that don't like water. That's why it's called hydrophobic. They hate water. And when these areas are exposed, a protein is not folded or unstable. So the chaperonin comes in, finds these proteins that are not folded, and begins to help them using its fuel, ATP. It helps to take that protein and bring it to a conformation that now is stable. All those hydrophobic pockets are now hidden, and now it's in a stable form that now it can do its function, whatever its function is help cells grow or help cells migrate or whatever, help cells make more metabolics that they need. That's what the chaperone does. But it also has a very important activity in addition to actually the mechanics of folding a protein. It's also a quality control mechanism. So it will actually make sure not only that the proteins are folded, that they're properly folded in the right shapes. So it comes through and says, yeah, you're good to go, you're finished. So this is what our chaperone does. And it does it in every cell of our body, but it does it more so in cancer cells. And here's why. So what we found, this is actually data from our lab, that in a healthy cell, you're going to find the chaperone. It's going to be there. But it's only folding about 1% of all the proteins in that cell. 99% of the other proteins are folded by other stuff. It's only responsible for about 1% of the proteins. If you look at a cancer cell, not only do they have more chaperonin, they make more of it, it folds more proteins. And these proteins are all the proteins that cause cancer. They're all the proteins that are coming off these mutated genes. The proteins, that's what the chaperone is folding. So this is a data I showed you guys last year, but it kind of makes the point. If you looked at all the cancers in the world, right, and you compared all the adult cancers, all the children's cancers, and normal tissue, and you asked, how much chaperone do all these guys have? You find that normal tissue has very low levels, that's the purple one. But as you go into adult cancers, those levels begin to increase. And as you go into pediatric cancers, it's even higher. And why is it higher in children's cancers? Well, it's because children's cancers are usually detected very late. They're not early stage cancers, while adult cancers are a mix of early stage to late stage. And we found that the chaperone actually increases as the cancers get worse. So it's logical that we'd see higher levels in pediatric cancers. But this is what we've been finding. And I'm going to go through this part quickly because I want to get to the new data. So if you look at across different cancers, so the other really exciting thing about the chaperone, it's not specific to one type of cancer. We have found chaperone in multiple cancers because it's a common pathway. All cancers need to fold proteins. And so we find it's elevated in breast cancers, especially as breast cancers move from early stage to late stage, and it's almost undetectable in the normal breast tissue. Prostate cancer, same thing. Almost undetectable in the normal prostate, high levels as prostate cancer increases in stage, and Gleason grade is a way that we measure severity of prostate cancer. Gleason grade 5 is among the highest, most severe aggressive forms of prostate cancer, and that's where we find the highest levels of our chaperone, is as the cancers become more aggressive and progress. And then neuroblastoma is a childhood cancer. And again, we found that it's actually a cancer not of the brain, it's a nerve cancer. So it actually occurs throughout the body. It's found everywhere in the body. And normal nerves, very little chaperone. But when you start getting into the higher stages of neuroblastoma, brown means stronger staining. You start seeing, again, that increased expression of CCT. So we've been seeing this since our initial studies in 2016 to our last studies this year. We've seen this increase of CCT in all cancers. Now, I'm going to share with you a little bit more of what I call bioinformatics data. And the reason is one of the great benefits that we have nowadays is we have access to thousands and thousands and thousands of cancer patients' data publicly available, which is really exciting. And so when I first started the Cure Bowl 10 years ago, it was really hard for me to prove what I just told you, that CCT is highly expressed in cancers, because I only had my lab's data to show that. Nowadays, I can go to a public database, take advantage of the power of thousands of labs doing research on their proteins of interest or their genes of interest. But because they're looking at whole genome data, they also have data for me, although they're not looking at my target. But that data for my target is embedded within all of their data. So this is an example. We were able to look at 19,000 patients with cancer and ask, how many patients had high levels of CCT in their tumors, and how many patients had low levels of CCT in their tumors, and how many died. And this is the trend. This is survival probability goes up on the axis here on the left, and the number of days is in the bottom here. And you can see that people whose tumors had cancer with high CCT levels did not do well. Their survival probability was much lower. And in fact, they all died. All these patients who had high CCT all died from their cancers. The patients who had cancer who had low levels of CCT, they were still alive. Most of them survived and did way better than their counterparts who had high CCT. So this is looking at all types of cancers, not just one single type of cancer. But if you focus on, for example, breast cancer, when I first did this analysis, again, 10 years ago, I think we only had a few hundred specimens that we could look at. Now we're looking at 7,400 patients' data, and we see this amazing trend. The difference between patients whose breast cancers are low CCT compared to high CCT is 5 years. In other words, people who have high CCT die 5 years sooner than patients who have low CCT. That's pretty impactful. I could not believe those numbers when I was looking at it. So this is telling you how important the chaperone is in cancer. And I'm going to stop here. Any questions so far?
I do have a question.
Yes, please.
Sorry, I'm going to be having to go pick up my class in just a second. First of all, thank you for the presentation. Very impactful with the numbers that you gave. Very exciting with all of the stuff. Is this going to be recorded? I believe it's going to be recorded. Is there an opportunity to see the rest of this presentation, even though I'm leaving? Is there a way that I could get a link to this later on?
I believe so. Is that correct?
Yes, that is correct. It lives on our website, David. Lives on our website. Last year's is on our website now, and we're getting ready to replace it with this one today.
That's fantastic. My father-in-law, unfortunately, we're going to have a service for him. He just passed away. He had lung cancer, but he survived 6 years. 6 years ago, St. Patrick's Day, he was diagnosed with stage 4 lung cancer. Went through immunotherapy. I think it was Keytruda.
Yes,
I believe
Keytruda.
it was the time, and he survived 6 more years because of the research that has been done, and that's one of the drugs that made it through clinical trial, which we were talking about. So he just passed, but your research is doing awesome, amazing things, and continue doing that. I look forward to speaking with you in the future and continuing on with the Cure Foundation as well.
And please, if you watch the rest of the video, reach out to me with my email. I'm happy to answer any questions. And I'm glad your father, Keytruda has saved the lives of so many people.
Yes.
I wish we could make it a cure. I
Yes,
wish we could turn that into something that cures lung cancer, right?
Yeah,
Not just a few more years. Yeah.
Yes. Thank you so much.
Thank you.
I appreciate it. Thank you.
Thank you, David. Anyone else? All right, Annette, the floor is yours.
Okay. So mining this, we came up with some really amazing trends as well. So this is now not asking just for survival, but now we were asking, well, is disease worse? Now, I had expected this. I was based on my observations. I had assumed that as cancer progresses, it becomes a worse disease, it becomes more invasive, we'd see more CCT. But that, again, was only the work from my lab. I didn't have data outside of my lab to show that. Now I do. So this is actually data from, again, looking at breast cancer patients. And breast cancer can be staged, and we'll talk about that in a moment, but it also can be molecularly typed. So we can actually look at the genes of the different types of breast cancer and break up breast cancer into subtypes. And the subtypes are luminal A, basal, HER2, and luminal B. And generally, luminal A has more favorable outcomes. So patients who have luminal A types of breast cancer do better than patients, for example, who have basal or HER2 enriched breast cancer. Those do worse. And what we found was that in orange here, the one on the far left, that's normal tissue. Again, levels are low. It increases slightly in luminal A, but really begins to increase when we start looking at the more worse prognosis cancer. So it's the types of breast cancer that have worse outcomes are the ones that have higher CCT levels. As I said, we now have data from thousands of patients that actually are showing that, not just what I did in my lab. Prostate cancer also has a similar trend. So prostate cancer is a little different from breast cancer. Prostate cancer is a very slow progressing cancer. So usually when it's detected in the early stages, it's treated quite readily and usually 99% survival in the early stages, much like breast cancer in the early stages. But it could take many years before it actually, if it does progress into a metastatic form, it takes many years for that to happen. So this is looking at individuals who did progress. So individuals whose cancers recurred and came back. That's the ones in the red. And the ones in the purple are the ones who didn't have it. So if you look at the very first one, it says CCT, that's our chaperone. And you can see that in the purple, it's very low. So patients whose cancers didn't come back had very, very low levels of CCT in their cancers. But those patients whose cancers did come back, did recur, had very high levels in the recurring cancer. So again, showing you that trend. Cancer gets worse, CCT goes up. Now, these other HSP90, AA1, AAB1, all this other, these are the heat shock proteins I was talking about in the beginning of my talk, and that they actually have treatments for. And if you look at the bioinformatics data for those, it's really bad. There is no difference. As a matter of fact, sometimes even goes backwards, where it's lower in the regression than it is in the original tumor. So the heat shock proteins don't show this pattern of worsening expression or increased expression with worsening disease, at least under this scenario. And again, this is data looking at thousands of patients. And then the other piece that I want to finish up is metastases. I had always suspected that we would see the highest levels of the chaperone and CCT in metastatic tumors. And metastases can occur all over the body. So this is something where you can, the tumor, for example, either begins at the breast or the prostate, it could spread all over the body when it's metastatic. So what this data shows here is comparing the CCT levels in the original tumor, which is the localized, that's in green, and the amount of CCT that's present in the tumor when it became metastatic. And this is looking just at prostate cancer. And you're probably asking, where's the green, right? Well, the green is way down here. In prostate cancer specifically, the levels of chaperone in the localized prostate is actually really low. When the prostate it hasn't spread, when it's in very, very early stages, it's actually low. It's higher than the normal prostate, but it's not as high as when it becomes metastatic. When you start looking either at lymph nodes, liver, when it starts to spread throughout the body, those levels of CCT just skyrocket, 30 to 50-fold changes. And again, this is not data from my lab. This is data mining all the data that's out there right now in the field. So again, showing that trend. And it's linked to death, because what we're finding, again, looking at survival probabilities, this is in prostate cancer, these are the patients that had high CCT, and they're all passing away and dying much faster than their counterparts that have low CCT. So again, the data is all coming together. And I was very happy, as I said, to see this data sets, because we have been seeing these trends in the research that was coming from my lab, but now we can show the bigger data sets that support what we've been seeing. So quick summary, and then I'm going to jump into new data. So what I want to do, and what we've been focusing in my lab to do, is develop a first-in-class drug that inhibits CCT. That's the big picture. That's where we want to go with this. And our reasons is, as I showed you, cancer cells need protein folding much more than normal cells do. So there's a dependency on that can be therapeutically exploited. We also know that all cancer cells need CCT. I showed you right now data from 3 different types of cancer, from a pediatric cancer, breast and prostate cancer. They all need CCT. And so we don't need to screen people ahead of time. We can directly move them once they have a diagnosis of recurrence, and this is where we're thinking that the best use of our drug would be. Once there's a diagnosis of recurrence, we can move into using a CCT inhibitor on those patients. And hopefully, as I show you in a moment, block the products from these abnormal genes that are causing their cancer. Again, it's highly expressed. I just showed you that data, so there's a therapeutic window. And as I showed you finally right now, we can target where the need is the greatest, which is the metastatic patient. we can actually try to help those individuals once the cancer has moved and spread throughout their body and they're not responding to the drugs that were working before, we can move in with our treatment. So this has been on my mind as I was putting all this together is, as I told you in the beginning, 95% of cancer drugs, clinical trials never get approved. And I really want to make sure that when our drug, when our CCT inhibitor gets to that point where we're now testing it in people, that we're going to be in the 5% and not in the 95%. So to do that, we have to create a large body of data to explain what is our chaperone CCT doing in cancer. Because if we understand how it works, what is it doing, we'll be able to design the clinical trials in a fashion that will lead us to success. We'll know the patients that will best benefit from this treatment. And that's the key to being successful in the clinical trial. So what we've done is we've taken cancer cells, and I'll show you data from our 3 cancers that we've been studying in the lab, and we've either artificially taken out the CCT from the cancer cell, or we've added more CCT to the cancer cell. And we've looked at what happens when we do that. And again, we've been focusing on that CCT2 subunit. So you're going to see the data that I'm showing you now is all in CCT2 subunit. The way we do it, I'm going to go very quickly over this. This is a method of depleting the CCT from cells, the chaperonin. It's called RNA interference. And basically all we're doing is we're removing the RNA for CCT2 from all the cancer cells that we're treating. So when we remove the RNA, it can't make the protein and the chaperonin isn't put together. So there's no chaperonin basically in these cells. And when we want to add more CCT, all we do is we add more DNA for CCT2, and that cancer cell will then make more DNA, more protein, and there's more CCT in that cancer cell. So again, just some nomenclature, we're going to look first at prostate cancer. So prostate cancer has multiple stages, showing you here. But the terms that I want to kind of tell you the difference is hormone sensitive versus castration resistant or hormone resistant. And what this is when prostate cancer recurs, what it does is it goes from a stage where it's responsive to the depletion of hormones, which is a therapeutic approach, and it's responsive. So that's what's called hormone sensitive prostate cancer. And then when it no longer responds to this treatment, becomes resistant to the hormone ablation therapy, it becomes castration resistant or hormone resistant. So you go from the stage of being sensitive to being resistant to the hormone ablation. And of course, the resistant form is the more serious, aggressive form of prostate cancer. So we did this collaboration with the Orlando VA. I'm going to take a little bit of time here because this is my exciting data. What we did is we looked at 20 prostate cancer patients. All these 20 patients had metastatic prostate cancer, but 10 of them were castration sensitive. They were still responding to therapy, basically, and 10 of them were castration resistant. They no longer responded to therapy. And what we found, if you look at the first column, it says pathogenic mutations. We found that as a patient's cancer moves from being sensitive to treatment to being resistant, the number of mutations in those tumors increases, doubles, in fact. You find there's an increase in mutations that occur during this period of time. So that is telling me, hey, these more unfolded proteins, right? More mutations, more unfolded proteins. So then we looked at what were the actual mutations. And so you don't worry about the details, just look at these 2 columns. So this column here says M1 CRPC. That's the 10 patients who were resistant to treatment. And they all had a different mutation. Each one of these numbers is a different patient. And every single patient had a different mutation. So if you think about it, if you're creating a treatment for these patients, you have to create 10 drugs to treat these patients. So you have to identify the mutation, which they did here, but now you have to match it with a drug, which means you need 10 drugs for 10 patients. So what we did is back up for a second. What we did is we took prostate cancer cells, we inhibited CCT using RNA interference, and our very expensive experiment, again, thank you Orlando Sports Foundation for supporting this, is we did what's called RNA sequencing. We took all the RNA inside these prostate cancer cells, we had depleted the chaperonin, and we analyzed each single one of them to identify all the changes in the RNA that occurred on every single gene in these cells. It's a global analysis. We got tons of data, but I want to point out the data. Let me back up for a second. So these are the 10 mutations. And I told you that you'd have to have 10 different drugs to treat these 10 mutations. They were all inhibited by blocking the chaperonin. So a chaperonin inhibitor, a CCT inhibitor, would actually be able to treat all those 10 patients because every single one of those mutations, this is the top row here. Red means high expression, green means low expression. So the top row is the untreated cancer cell that has lots of chaperonin. So this is like the baseline. They're making lots of chaperonin and they're making lots of products from these genetic mutations. The bottom one is when we remove the chaperone, the depleted one. So remember, high is red, green is low. Everything here is green. You see how they're all green? Every single one of these gene products was reduced. So again, telling you we can treat all those 10 patients with a chaperone inhibitor and they would be able to have reduction of their tumors. And the point is the chaperone, because it can block multiple targets, it's a multi-target approach versus a single target approach, which is what the current drugs are doing. So let me stop here. Any questions so far? Okay, so the experiments that we added more chaperone to were actually pretty intriguing as well. This is a prostate cancer cell that has the normal amount of chaperone, what they normally make, right? The basal amount, which is usually high, but the normal amount. We added even more chaperone. Here's what those cancer cells look like when they had more chaperone. The little green dots that you're seeing here are antigen receptors. So antigen receptor is a driver of prostate cancer. So you can see that there are green dots in these cells. But these cells, not only did they divide and grow faster than the cells without more chaperonin, they actually made tons more antigen receptor. And the red is actually the skeleton, the cytoskeleton. They were making more cytoskeleton. So these cells actually just took off. They were just growing happily when they had more chaperonin. So again, showing you how important the chaperone is to cancer cells. So briefly, we are currently working with the VA on expanding this data set that I just showed you. I just submitted a merit grant with them, so keep our fingers crossed that should be reviewed in a couple of months. Exciting news from the Bay Pines VA. They were so happy with the work that we were doing that they actually offered to provide some funds for us to do animal work at their facility over in St. Petersburg. So I'm currently working with them on doing a prostate cancer animal model that we've been developing in my lab, and they're going to fund part of that project in their facility over at Bay Pines. And we also submitted some new stuff, which I'm going to talk about at the end, to the DoD, and actually scored really well. We didn't get the grant, but we actually scored really well, and we didn't have a whole lot of data either. So I'm really optimistic this year that we'll resubmit to the DoD with now more data and hopefully get into that funding range, which is usually our goal. All right, so the next few minutes, and again, I think I have about 15 more minutes, Alan. Is that good? Okay. So I'm going to go quickly over the next two cancers and kind of show you where we've been heading. Breast cancer. We've actually been continuing our studies in breast cancer where we actually began all our work 10 years ago that we've learned over the last 10 years about what the chaperone does in cancer, all began with breast cancer studies. And, breast cancer, I told you, have these different types of subtypes where these HER2 and basal-like are the ones that have the worst prognosis. And that's the type of cancers that we're finding more of the chaperonin is being expressed. So we took a luminal A breast cancer cell line called T47D. We inhibited CCT2 in that, like we did in the prostates that I just showed you. We did RNA sequencing with these cells. And what was really amazing was, and I'm going to show you that in a second, is that we had a really global effect on these breast cancer cells just by knocking out one single protein of the CCT subunit. But briefly, what you're going to see in a second are signaling pathways. And what a signaling pathway is to a cancer cell, it's kind of like a lock and key idea. So if you think about it, the cancer cells have all these receptors on their surface, and these receptors are going to bind to different proteins that are found in the environment. It's called a ligand. So a ligand receptor interaction. It's like a lock and key interaction where the key is the ligand and the lock is a receptor. When they fit, that triggers a whole release of these chemical messengers inside of a cancer cell that leads that cancer cell to have some sort of response, whether it's proliferation, expansion, migration, survival, all these things that cancer cells do is driven by these chemical messengers that are made in response to some sort of receptor interaction. And one of the chemical pathways I'm going to show you is called the MAP kinase pathway. Kind of looks like this, where you have epidermal growth factor receptor that binds to its receptor, and then it triggers this whole cascade of MAP kinases, which are the chemical messenger. And the end result is making a transcription factor called c-MYC that causes cells to divide and proliferate abnormally. So this is like a typical cancer signaling pathway. So we took our T47D cells that were depleted for chaperonin, sent them out for RNA sequencing, and we got this data. And again, back to the same idea I showed you before. Red is very high expression, green is very low expression. And the three columns are, the first two columns are the control cells that have normal levels of the chaperonin. And the third column, in all the boxes I'm going to show you in a moment, that third column is always our experimental sample, the one that we knocked out or depleted the chaperonin. And again, green being low. And I think this kind of speaks for itself. You can see that CCT was our target, was almost 100% depleted in these cells with our depletion method. And the other subunits that make up the CCT complex as well. So those all got knocked out. We didn't target the other ones. We only targeted CCT2. But CCT2 is so important that when it's gone, all the other subunits also disappear. Well, we looked at MAP kinase signaling, and I'm just going to show you, just look at colors. Green, 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 green. Knock down all the MAP kinase expression. PI3 kinase, which is another signaling pathway, pretty much similar trend, except for one exception, which is a protein called AKT1 and 2. Those went up. So what we're seeing is not just a simple everything got knocked down. We're actually seeing abnormalities in the RNA patterns where some go up and some go down. So we've completely changed the functionality of this cancer cell. Wnt signaling is another very critical pathway for both breast, prostate cancer, and neuroblastoma, which is the last one we're studying. And the critical player here is the first one. It's called beta-catenin. Again, it's green. We knock down beta-catenin in all these cancers. And again, that pattern of, you can see that the pattern of colors is very different than the control cells. Now, molecular point, same thing with focal adhesions. These are the proteins that help cancer cells migrate and attach and spread. This is the key to metastases. We're knocking all those down as well. And cell cycling. These are the things that make a cell divide and proliferate. Again, knocking those down with one exception, which is CDK4, and that one went up. So again, we're seeing this up and down pattern of changing how these cancer cells are responding. So this was the first time we've actually been able to probe inside of a cancer cell and ask what happens when we knock out CCT? How does it globally change this cancer cell? So the last thing I was going to talk about with breast cancer was our newest direction, which is now we're looking at something called exosomes. And I'll tell you, exosomes are kind of scary when I first started to read about these. What these are, and you can see here this little picture that I've got. In purple is our cancer cell, and in green is a normal cell. And what the cancer cell is doing in this picture is it's actually releasing little particles made from itself, little particles into the environment to be taken up by other cells. And the theory that is in the field right now is that these little particles called exosomes actually contain the seeds of future cancers. So if cancer cells are releasing these exosomes, that's how they lead to potentially seeding new sites. Maybe a cancer cell is not necessarily moving from site A to site B to cause metastases. It's actually just shedding these exosomes that are picked up by normal cells at a distant site. And those normal cells get turned into cancer cells. That's the theory, at least, that the field has. So we wanted to look at exosomes because we were wondering, well, those cells that have more CCT, are they making more exosomes? Yes, they are. So this is a piece of data that we just got. This is very recent. This is showing you in the gray is the normal cell without a lot of CCT. And in the white box is the amount of exosomes released from the cell that had lots of CCT. So gray is no CCT, white is lots of CCT. And the number is what the y-axis has shown over here. Yes, they definitely are producing more exosomes. And those exosomes are producing actually have the RNA for CCT. So we're actually able to we isolate these little green balls that you see here. We isolated the RNA from them, and we try to detect CCT2 RNA in that group of RNAs that we pulled out. And what you can see here is that there's tons of CCT2 RNA. What does that mean? Well, that means that any cell that takes up one of these little green balls with CCT2 RNA in it has the blueprint to make CCT2 protein. So it actually can start making its own CCT2 protein based on getting that RNA from that exosome that it took up. And that it has an impact is shown in this last figure. This is a migration assay. And what this line shows is how fast are these cells moving. The red one in the middle is kind of the control. That's how fast the control cells move. When you add more CCT, that's the green line. These cells start moving really fast. They start migrating all over the place. And if you take away CC2, that's the yellow one, they stop moving. So imagine what these CCT containing exosomes are doing to cancer cells. So this is our new area of research. This is where we want to move into this year. We want to study these exosomes better and actually develop a diagnostic based on detecting these exosomes with CCT. And last but not least, and this will be, I'll go very quickly, this is on neuroblastoma. Neuroblastoma is a pediatric cancer that we've been focusing on. And we've been working on this cancer because it's actually cancer detected in infants, less than a year old. And the challenge is, it's as I said, it's a cancer of the nerves. And it usually appears in the early stages in the adrenal gland. And what happens is when it's actually detected, if it is, because often it's not even detected because it doesn't cause any symptoms initially. But if it is detected, there's nothing that a physician can do. They actually have to wait. So usually what happens is most of these tumors, when they first appear, many of them spontaneously regress. And a small number of them that don't regress are the ones that turn into these late stage neuroblastoma that spread throughout the body of the infant. But doctors don't know how to tell the difference. They don't know if that patient is either going to regress and absorb the little tumor, or the tumor is going to turn into something nasty. And by the time they've detected the spread, it's actually too late. It's already spread throughout the body. So one of the first things we did, we were funded from the Live Like Bella Foundation. This is through the Florida Department of Health. We developed a biomarker diagnostic assay that can be used with those infants just by taking blood, and we could detect those neuroblastoma cells in the blood. The piece of data that we didn't have was, does detecting these neuroblastoma cells in the blood mean that they're going to get aggressive disease? We didn't have that piece of data. So we had the assay, but we didn't have that piece of data. So that's what we're doing right now. And what we did is we did very similar, like I just showed you for breast and prostate. We took our neuroblastoma cells, we inhibited CC2, we sent them for RNA sequencing, and what we got was very similar data. Yes, we are detecting when CCT is removed, we actually lose all the functionality that these cells had of becoming aggressive cancer cells. So CCT is very important for these cancer cells. So detecting CCT containing circulating tumor cells in blood, like our assay does, actually does mean that child could potentially have the more aggressive form of that neuroblastoma. So that's where the study is. We got funding from them again last year, and we're currently doing some studies on developing a treatment. And this is the data that we just got. This is hot off the press data. This is us when we use our inhibitory approach of knocking down CCT RNA. We did this in a mouse model of neuroblastoma. And in this mouse model, the neuroblastoma actually occurs in the fat. So to show you a quick picture here, this is the inguinal fat of a mouse shown down here. This is where we injected the tumor cells. This kind of corresponds to the belly fat that we have as humans. And what we found was that the mice that had abundant CCT in their tumors developed these really huge nodules of fat depositions, basically. You can see here, these are all tumors growing in the inguinal fat in these mice. And in addition, 3 of the 6 mice also developed skin lesions. These are subcutaneous tumors. So the tumor was just growing everywhere in these mice. Now the mice that got, the lucky mice that got the CCT inhibited tumors, these are the tumors that didn't have CCT function in them, they grew very, very, very tiny tumors. You can see here, very, very small tumors. In fact, They even had less fat. And this is the amount of fat that was in these tumors. Oh, and by the way, we could not separate the tumor from the fat because the tumors were so embedded in the fat in the control mice, but we could easily remove the tumors from the fat of the treated mice that had the chaperone inhibition. So you can see here, very, very small. So it was almost, when we did the numbers, we almost had a 60% reduction of metastatic spread, not just tumor growth, but metastatic spread in these mice by just blocking CCT. And of course, we confirmed that the tumors, in fact, the control tumors had high levels of CCT, that's shown here in the black, and our treated tumors had low levels of CCT, so we confirmed that. But this is where we are now. We are moving towards developing our inhibitor. And our inhibitor is going to be a small peptide, which we've actually been working on for many years. We just didn't know what it did. So 10 years ago, we actually had this peptide in hand, but we didn't know how it killed cancer cells. So today I can tell you, we know how it works now. And so we're in the process of developing the next generation of these. And I'm gonna go quickly over this part because I wanna get to the end. We started working with nanoparticles. That didn't really lead us far because nanoparticles end up going mainly to the liver. They weren't very efficient at taking our peptide to tumors. So we kind of just stopped with the nanoparticle approach. We're not doing small molecules either because it's very hard to make a hydrophobic small molecule. And our peptide has to be hydrophobic to be able to inhibit the chaperone. And so what we're working with is we're actually using, go over here, we're working with salmonella as our biofactory for CT20. So this is our funded study that we're doing from the Live Like Bella Foundation. So we're making bacteria that make CT20 that will infect tumors. And so that's our direction that we're heading. I'm not going to go through the preliminary data yet because there's still stuff to do. I will tell you that we did have one drawback. I call it the leaking inducible expression problem. And when we're trying, we have to actually put the DNA for CT20 in the bacteria. So this is the process of doing that. You have to put it into a plasmid, you put the plasmid into the bacteria. And you have to be able to turn the expression of that CT20 off or on. Because if it's on all the time, everything gets killed and you have nothing to study. So we wanted to do an inducible expression where we could just turn on the expression of CT20 when we wanted it. So it's off, default is off, and we can turn it on when we want to. But the problem was the system we were using was on all the time. It was leaky. So we were not getting, so this put us back a couple of months of work trying to figure out why in the world we couldn't turn things off and on. But we solved the problem. It's now solved, yay. We actually have a two-step system now where we're actually putting in the first step, which is putting a lot of the suppressor so we can have this the off switch on the time. And then we got a second step where we had our inducible system, and now we can turn it on whenever we want. So as I said, that problem was solved, but it did put us back for a bit, but we're moving forward, hopefully. Where I want to go in the last 2 minutes is telling you about how we're going to make CT20 bigger and better. And this is where we're heading in the project this year. One of the things that I decided to do was take a look again at how CT20 inhibits the chaperone. And the way it does it, it's basically a reversible effect. Basically, it binds to the chaperone, blocks the ability of the chaperone to bind to ATP, the fuel basically of the chaperone, and then things happen, right? The chaperone stops working. But the minute CT20 is gone, the chaperone can resume functioning. So it's a reversible effect. It's not a permanent inhibition of the chaperone. So I really wanted to use CT20 to make a permanent effect and eliminate the chaperone completely from cells. So the way to do that is this method called PROTACs, and it's really cool. What it is, our target is the chaperone, right? That's our protein target. And what we want to do is we want to use CT20 to connect another protein that's called an E3 ligand. And think about that like a garbage truck. So we want to connect CT20 to a garbage truck inside the cells. And what that will do is that once we make this new molecule, CT20 attached to garbage truck, that will take the CCT, and that's shown down here in this figure, it will take the CCT to another compartment inside of a cell that's basically a garbage disposal. It's called a proteasome. And every cell has this garbage disposal where it eliminates protein it doesn't need anymore. So we're going to actually be able to take CCT, drive it to the proteasome with this E3 ligand or the garbage truck, and that will then cause the chaperone not only to be stopped functioning, will degrade and destroy the chaperone. So this is what we're heading right now to do. This is kind of what it looks like. This is our new CT20 molecule that we're making in the lab. It's actually it's like a little soap bubble. It's a micelle of multiple CT20 peptides all together with different functionalities. It exists as a glob, if you think about it, outside the cell. And then when it goes inside the cell, it breaks apart into individual peptides. And some peptides do this garbage disposal mechanism where it destroys all the CCT components and there's no more CCT. And some of those will come in and do that inhibitory effect to any preformed complex. So we can actually hit cells two ways. We can hit all the preformed chaperonins and block their activity, and we can prevent any new chaperone from forming with this garbage disposal method that we've added functionality to. So I'm really excited. So we got a Superman now. Our CT20 has turned into a super CT20, and that's what we're testing. We have no data yet. This is just simply the ideas that we're doing. But I have a colleague who's actually synthesizing the CT20 with the garbage disposal attached to it. And I hope to start testing those in the lab really soon. But this is where, this is where kind of just leave you with this thought. This is the current dogma. Genetic testing linked to targeting, to matching with drugs. And as I said, in the example that I showed you with the prostate cancer patients from the VA, you need a different drug for everybody because everybody's tumors and genes are different. We can treat everyone with our inhibitor as long as their cancers are progressing and the CCT levels have increased. I don't see this as a treatment for early stage cancers. I think we have very effective treatments for early stage. Where we have a need is in the metastatic and recurring cancers, where patients no longer respond to the treatments that they had, and doctors are now just scrambling. Every year they have to replace. And this is talking to an oncologist. She told me that most patients, especially in the prostate cancer world, they will respond for about a year to their treatments, and after that they'll stop responding. And they have to change people every year to a new treatment. And after a while, they run out of treatments. And so I'm hoping that our approach can actually put a dent into this problem and really provide an alternative for oncologists to help patients live longer and maybe even in some cases be a cure for cancer down the road. But I'm optimistic. We're also developing the exosomes, which I mentioned. And I will finish off just thanking the amazing people in my lab who are doing this work. James, Colton, Carolyn, and Lamb, they're all students at UCF, and they're all doing this work that I showed you. So this is done by undergraduates and graduate students in my lab who are working on this. And again, thank you all. I am so excited. I'm looking forward to in the coming year, we're going to be moving, if all goes well, to the Lake Nona Cancer Center. You all will be visiting the Cancer Center hopefully later this year. And I'm really looking forward to start bringing together the teams now in our new location. I will stop here and take any other questions that you guys may have.
Yeah. Does anybody have any questions for Annette? Please, please.
Doctor, I just want to say I am so appreciative for such a brilliant person like you who can talk at a level that common people like myself can understand. This is the best presentation and given the best understanding I've ever had. Going back and laying the foundation, just the simplicity of what is cancer is huge, because most of the time people talk at a level that's already way above. And thank you so much. You're just so practical, and I appreciate everything you're doing. God bless you, and praying that God will do a miracle through you guys this year as you continue to progress with what the insight he's given you to be able to develop these things to be used around the world. Thank you.
Well, thank you. You made my day. Thank you for your comments.
Anyone else? All right. First, Annette, I wanted to, before we dismiss you, dismiss everybody, we want you to hang on because we'd like to get some sound bites here on the research. But even an old football coach, I'm taking my notes here, and I'll quote Jamie, or on top of his comments. We really appreciate the way you do it. But for all of us to be able to go out and tell our stakeholders in a compact statement, an executive summary of your research this year, which to me was amazing, that your research has created a CCT inhibitor through your RNA sequencing research That's the big step forward. Am
That's
I
the big
right?
step. Yes, that we've shown that through RNA sequencing data that we are definitely impacting tremendously these cancer cells, that we're changing their entire scope, right?
And it doesn't matter where the cancer is, what tumor.
Yeah.
All of them are still.
And they all change
contained.
differently. This is the amazing piece, you know, it's not that they all change the exact same way. They all change different ways, but every change caused them to die.
Yeah, that's great. All right. We appreciate everybody's support. Appreciate you coming out today to hear Dr. Khaled. You know, this is really what it's all about. Anybody that you come in contact with, I know everybody here that's on this call is involved with the Orlando Sports Foundation, our events. Why do you become a part of this? You're bringing teams together to find a cure for cancer. And they can go to our website and see where the money is going. You know, this is real, and we are trying to solve cancer through Dr. Khaled's research, and she just needs more money, you know, and we're working through all of our events, all the sporting events that we have to create those funds for her research. So has a lot of credibility what we're doing here in the Orlando Sports Foundation, and we appreciate you all. Annette, if you'll hang on, everybody else, thank you so much. Go to our website and check it out if you need some more details. Thank you again.